From Microbe TV, this is Immune, episode number 26, recorded on November 26, 2019. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello again.
Welcome to November.
Yeah.
Pretty soon the year will be over.
Don't say that. I'm not ready for that
yet. I am looking forward to 2020. What a great year number, right? 2020.
Yeah, it's true. It's true. There are some reasons I'd like to be done with 2019. So yeah, let's get a move on.
Also joining us from Durham, North Carolina, Steph Langel.
Hey there. Good to be back. It's a nice fall day here in Durham. Not too hot, not too cold.
Do you have fall? Do you have fall in North Carolina?
It took a long time, but it finally came.
Did the leaves drop?
They're still on, and they're colorful and it's beautiful.
Today we have a guest from Boston, Massachusetts. He is at the Harvard School of Public Health. Michael Mina, welcome to Immune.
Thanks so much. Keeping with the theme, it's a beautiful day in Boston today. Much nicer than a few days back.
Yeah, you can get cold, nasty weather up there. I know. I spent a couple of years there as a postdoc.
Yep.
And so, and it happens sooner than down here.
That's for sure.
So we're going to talk about a very cool paper that Michael and his colleagues published today. Before we do that, let me remind you that if you like what we do here on Immune and all our other podcasts, consider supporting us financially. We could use your help. Microbe.tv/contribute. We'll have more on that later. So, Michael, tell us a little bit about yourself. Where are you from?
Sure. Well, I'm currently at the Harvard School of Public Health, where I'm an assistant professor of epidemiology and of immunology and infectious diseases, and I'm also a physician just next door at Brigham and Women's Hospital and Harvard Medical School in pathology. And I actually grew up in the Northeast and spent quite a while down south in Atlanta at Emory and then at Princeton, and worked my way back north again, where I'm up here at Harvard now.
So you're an undergrad at Emory?
No, I was an undergrad at Dartmouth. I was an engineering undergrad, and then I did my MD and PhD at Emory, and sort of most of my PhD was actually at St. Jude Children's Hospital, and then I started working my way north again after finishing med school at Emory.
What did you do at Princeton?
I was a postdoc at Princeton with Brian Grenfell, and that was actually where all of this research on measles for me really got started. Brian is a mathematical biologist. He is he's been working on measles dynamics and understanding why and when measles outbreaks happen for pretty much the vast majority of his career. And so I was doing a postdoc with him back in 2013 and 14, which is how I got into the measles world. Before that, I was working on influenza and bacterial co-infections, which I still continue to work on here and there.
And that's an interesting time to get involved in measles, right? Because that was when we started having outbreaks again here, especially.
Yeah, exactly. So it was 2000. I mean, it was really in 2014 15, right? That I was. Yeah, exactly. So there was the Disneyland outbreak in 15, and before that, I felt like it was odd because I think people in the measles community felt like they were having to justify the fact that they were continuing to work on measles, and then, of course, measles didn't, in much of the world, measles never really went away. But in the States, it came roaring back with, into the public mind because of the Disneyland outbreak, and it sort of continued to only increase from there, unfortunately.
Right. It's that paradox between, of course, we don't want measles, but now I imagine there are more measles funding opportunities.
Yeah, it's all of a sudden it's become a sexy thing to work on measles again, which would have been really nice if that didn't happen. But
right, before the outbreaks.
Yeah, exactly.
So, give us an idea of the global burden of measles at the moment.
Yeah, so measles. Probably most people, I don't know who your listener base is, but most people listening probably don't see a lot of measles. But it still is quite common in much of the world. There's about 7 million, back of the envelope calculation would suggest around 7 million or more cases continue to happen globally. Much of it is in sub-Saharan Africa and parts of Asia. But of course, in the recent years, it's come roaring back into Europe. There's been tens of thousands of cases in various countries, totaling hundreds of thousands of cases in Europe now. And this was a disease that just not too many years ago, we were talking about, full-on measles elimination and potential for it to be the next pathogen that's eradicated. And of course, those gains are clearly being are kind of falling back now. But it's still a disease. It ends up killing. The WHO estimates, Henry, that it costs the lives of over 100,000 children globally each year. I think some of the work that our recent work and past work from a few years ago suggests that number is probably even considerably higher than that in a less direct way. And so it's a serious infection. In the recent outbreaks, of course, there was the Jewish Orthodox community in Brooklyn and the New York City area, which led to almost 1,000 cases, and across the country in 2019, I believe there has been over 1,000 cases now, and almost 20% of those cases, and this is in a place like the United States where kids are well nourished, and but still about 20% of the cases are landing kids in the hospital and in the ICU. So it's a really serious disease that is unfortunately coming back into the public attention.
And we're talking about increases here from almost zero circulation in the U.S., right? Because weren't we declared eliminated in 2000? Is that right?
Yeah, that's exactly right. And that's where it's just so astounding. I mean, there's always been, elimination status is really based on the elimination of continued chains of transmission for a year. Right. And so over the past 2 decades, there have been small little outbreaks from somebody coming in from another country and bringing measles with them, and maybe it infects a couple of kids. But this is really, at the national level, it's been almost zero cases, for the most part. And we were considered eliminated. In fact, measles was eliminated from all of the Americas, North and South America, together in the early 2000s. And of course, now you're having huge measles outbreaks in Brazil, all over the Americas. And so it's really an astounding rebound that's occurring, and much in particular in the United States, the problems are a combination of course, increasing vaccine hesitancy and actual increasing anti-vaccine rhetoric. And I do make a large distinction between the two. But when you couple that to increasing outbreaks that are due to political strife and breakdowns in immunization policy and practice in developing countries and other countries that are neighboring the U.S. or in Europe, you now have huge outbreaks that are happening all around the world with increased travel. Those individuals are coming into the United States, and when you couple that with sort of vaccine hesitancy and an enormous number of people now who are choosing to not vaccinate their children, it's leading to the situation today where we only, the future, at least the near future, looks bleak. I suspect that we're going to continue having larger measles outbreaks here.
Right. And so before kind of the vaccine hesitancy being a reason potentially for the increases, in the areas of the world where the measles is increasing, was it because of lack of infrastructure and a break in the chain? Or like rotavirus, or the rotavirus vaccine, is it less susceptible in those areas because of micronutrient deficiencies or other reasons? Was there evidence of that?
Yeah, so there's not a huge body of evidence for measles anyway that micronutrient deficiencies are leading to increased susceptibility. Measles is sort of this virus that it usually attacks anyone who's not been immunized or infected in the past, and whether you're sort of have other like lower socioeconomic backgrounds that lead to nutritional deficiencies, that really doesn't impact on whether or not you'll be susceptible to measles, but it does impact greatly on what your course of measles will look like. And we know that there's a very strong link there between, for example, vitamin A deficiency, which has been spoken about, surprisingly a lot in the vaccine hesitant community lately, and we've been and in the professional societies as well. But nutritional deficiencies will lead to much worse measles, but I think it's a little bit different. You won't see increased susceptibility per se. Because
what about the seroconversion of the vaccine? I mean, with the vaccine specifically?
Yeah, so I think the vaccine. So there's not there's a lot of questions surrounding what's going on with. We do see that it looks like there's increasing numbers of people who are getting measles even though they've been vaccinated, and some of that's because of just breakdowns in the vaccine program, leading to only one. A lot of individuals are maybe only getting vaccinated once, and they're not getting their second dose, which is really necessary for sort of what we consider often to be sort of full protection. In that case, it could be. I mean, I would say that there's not any good data to suggest who is really breaking through and who's having these secondary vaccine failures, they're called, when you've been vaccinated but you end up getting the infection. And there's a lot of talk now, is it because of antibody waning and protection is waning because potentially because there's not natural exposure is also sort of giving people sort of natural boosting. And but I don't know that the nutritional status has been fully explored at this point to suggest that that's really a driving force underlying sort of secondary vaccine failures.
I would guess that most Americans have no idea that there are over 7 million global infections currently.
Right.
Yeah, it's considered. Most people haven't seen it. They think that it's just a benign childhood infection, and it's a huge number of infections that still occur every year.
In fact, that's what a lot of people who don't want the vaccine use as an excuse. They say, "There's no more measles. Why should we get vaccinated?"
Right.
Yeah, it's
crazy.
Yeah, you see it. It's definitely a moving target for sure. I mean, not that long ago, it was the major thing was really this fear that has now been completely debunked, that measles vaccine was leading to autism and these kinds of things. And then it's definitely a moving target to the point where it's been really getting hearing what a lot of the concerns are now. Now people, it's gotten so far that most people just want to pretend that measles is just not bad for you. And it's actually going in the other direction where there's a growing body of people who are really putting out there that measles is actually good for you and that kids should be getting measles. And it's just a really, it's a dangerous.
Really,
so crazy.
I heard that death is good for you. Is that right?
Yeah.
Unbelievable.
You won't get sick again.
Yeah.
Well, that's true.
I mean, these 100,000 plus deaths, what typically is the cause of death in these countries?
So with measles anyway, there's the acute phase of measles, and sometimes you actually have individuals die of a viral infection itself. But usually that's not really it's usually a secondary infection that's happening in the very acute phase, right during or after measles. And that's because right when a child is actually infected with measles, it's highly, highly immunosuppressing. It shuts down your functional capacity and it shuts down your overall defenses at the same time as it causes your epithelium to be more susceptible to bacterial outgrowth and various other infections. And so you actually get a lot of bacterial pneumonias and diarrheal diseases that end up causing a lot of the death that we end up seeing. That's associated with measles infections.
So correct me if I'm wrong. So far, there has not been in the recent outbreaks in the U.S. anyway, there hasn't been a measles-associated death, right?
That's correct. It turns out we're extremely good at keeping people alive in the hospital in the U.S. Yeah.
Yeah. Has there been any encephalitis yet?
I don't know of any in the current outbreaks that I've heard of. Of course, I mean, the expectation is there might be some SSPE. This is these long. These long-term problems that are associated with measles, where you end up getting encephalitis 5, 10, 15 years after an infection. And I wouldn't be surprised if we do see some cases a number of years from now that are really associated with this current outbreak. And in fact, I think a number of years we did have a few cases of SSP that were linked back to a measles outbreak, I think in 2005 or something along those lines. So that's the scary thing. You never know when it's going to come back, and some of these viruses can be harboring away in our brains, and we don't know it.
An SSPE is
subsclerosing panencephalitis, and
okay.
So it's an encephalopathic process that occurs and causes an encephalitis, and it's almost always lethal. I think it maybe has 100% lethality, but it.
And how would they know that that's associated with prior measles infection?
You actually on autopsy or in biopsies, you can actually pull the virus out.
Oh wow!
Yeah.
So it stays, because this is not a DNA virus, right? so it stays around.
Yeah, how it ends up happening.
How does
in the
that
brain
work,
is,
Vincent?
Well, as Michael said,
Flexing.
so you can find viral genomes, although you don't find infectious virus.
Right.
Well, you find viral genomes, you find proteins, viral proteins, and how it persists and why it causes the illness is completely unknown.
Wow.
Very difficult. It's very rare, and there's no animal model, so can't do anything. The best thing we can do is vaccinate, and that will prevent it.
Yep.
Yeah.
So, these things this is just logic, and I just feel we're in an age of disinformation in the world, and there are certain people out there who want to foment disinformation and discord, as we know, and this is, I think, part of it. It's not just political stuff, but it's also healthcare and other aspects of our lives that they. Every time I argue on Twitter with someone who's not interested in vaccinating, I always wonder if they're a Russian person who is being paid to do that, spread disinformation.
Right.
Anyway.
Yeah, I actually I tweeted out this. I'm pretty new on Twitter, and I tweeted out a tweet, I guess, the other day, maybe 2 days ago, and it's gone a little bit, mildly viral in my little Twitter world. But it was based on an amazing conversation I had with some anti-vaccine folks on Twitter, and there's been a lot ever since the paper came out a couple weeks ago.
I would imagine so.
Right.
And but the amazing thing was I was advocating for vaccines, and the response I got back from this individual was, "Oh, and do you consider yourself an expert on vaccines or immunology?" And I said, "Yes."
Ew.
And the response to me saying yes was, "And you don't think that's a conflict of interest?" And
That's interesting.
that's kind of where we're at now.
Right?
Yeah, experts don't mean anything.
Right. If you're an expert, you're clearly conflicted, and you're giving just the information that the big pharma companies want, because clearly they're, it's really unfortunate. And I think there's a lot of misinformation. It would be a really interesting, maybe at some point you could do a whole show about, maybe you've already done it. How's research funded? And I think there's a lot of misinformation about how research grants actually get funded. It's not sort of the government and pharmaceuticals coming to us and saying, "Oh, please do this research." It's us begging them for some money, and
for
begging
years.
is the operative word there.
Well, one person said to me, when the companies that make vaccines are not involved in their testing, then we can have a conversation. It's like, well, what do you expect? Someone else, a third party, to test someone else's vaccines for you? I mean, to a certain extent, we have clinical trials that do that, but they're always run by the company who makes it. There's no other way to do it.
Yeah, it's like an untestable hypothesis in science, right? You come up with something and it sounds really good, but there's no way to test it. And so it's coming up with an absurd argument that's undoable.
Right.
So I happen to be reading this book by Rachel Maddow.
She's
I think it's mostly about the oil and gas industry, but it's also about the disinformation campaign that the Russians have engaged on for quite a few years now. And now I think everything on Twitter and Facebook that's contrary is one of them. I'm paranoid because just think about it. Like this person that was talking to you, Michael, they just want to sow discord. and make unreasonable claims. And, I found whatever you say, you're always wrong.
Right.
Now, for our audience members who may not understand why we do need pharmaceutical companies to get a product to market, and the biggest reason is because it is a $1 billion investment to get something through clinical trials and to a product on market. So, I mean, I guess there could be a way that could be taken up by the government solely, but that would be a huge,
But they approve it in the end, right? You give them your results, you file it with the FDA, and they say yes or no, or go back, right?
Correct.
Mm-hmm.
And then if you tell these people that, they will say, but the FDA is involved too, right? Everybody's involved.
It's very,
it's a huge conspiracy. Every physician and every researcher and everyone's out to get the world.
Right.
Conspiracy. In my experience, you can't get 5 scientists to agree on anything. The idea that they would have a conspiracy is so crazy.
Very
That's
funny.
true.
Anyway, let's talk about your paper.
I was going to say, I assume you didn't get into this to get into to the anti-vax argument there, but you probably got into this from what we started off talking about, is that this is causing these deaths, and as you said, the deaths after measles are often not from the measles directly, but from these other infections. And is that what spurred you to go and look into what you're doing? And would you tell us the title of your paper and what your focus was?
Sure. So, I'll just tell you. I'll start off by telling you that the title of the paper, and actually, it's kind of a long title, so let me just pull it up here so I don't get it wrong. It sounds ridiculous, I know. So the title of the paper is Measles Virus Infection Diminishes Pre-Existing Antibodies That Offer Protection from Other Pathogens. So put shortly, it's essentially that measles virus, when somebody gets infected, it seems to delete or erase some of the pre-existing immunological memory. And that can ultimately cause somebody to be more susceptible to infectious diseases that they should have previously had good protection against. And so what actually got me into this work was this was published in Science just a couple of weeks ago, but in 2015 we published a paper that actually is quite similar in a way, but totally different in terms of the methods and the actual findings. But that was also published in Science, and that was essentially, I think the title was that measles-induced immunomodulation increases childhood infectious disease deaths. And that paper was really based on the idea that if measles infections, if measles virus can actually infect lymphocytes and immune memory cells, could it be increasing risk of other diseases? And so taking a big step back, what really led to this is that ever since measles vaccines were introduced in the 1960s, first in the U.S. they were introduced in 1963, and then in the U.K. they were introduced in 1968, and then were kind of rolled out across the globe after that. There have been really remarkable reductions in mortality that were seen in the years after the measles vaccine was introduced to any given country or region. And I think I've looked at a lot of different observational studies, and these are from all different people around the world. There was one study, in fact, in Haiti that showed a 90% reduction in the overall childhood mortality after measles vaccine was introduced.
Wow.
And that was an extreme case, and that was a small, very, very impoverished community. But so these observations were made, and there's been a lot of work to try to understand them, and most of the hypotheses have centered on some sort of boosting of immunity overall based on the jab from the vaccine, and with the idea that maybe a kid gets a vaccine and it somehow stimulates the immune response to sort of be boosted for a number of years. I think as an immunologist and physician, it's kind of hard to swallow that and believe that the vaccine itself is somehow really turning on the immune response for years.
Right.
Now, there is some interesting work from Miha Netea in the Netherlands and a number of other people, epidemiological work from Christine Ben and Peter Abbey that are suggesting that maybe these effects do exist to some extent. But the effects have just been so incredibly strong for measles vaccine that it led us to put together a different hypothesis. And the other hypothesis was really born from very nice work in macaques, in these non-human primate models, where Rick de Swart and Roy DeVries and a number of other people, as well as Diane Griffin, who's at Hopkins, have shown over the years really nice data that shows that measles virus infects lymphocytes and can infect huge numbers of lymphocytes. And we can track all the cells that get infected. And this is actually Paul Dupré also has been very, very instrumental in all this work. And essentially the idea is to put a GFP fluorescence protein on a measles virus, and then that allows you to track where that virus is going inside of a primate's body. And what that work showed was that at any given time during acute infection, you might see up to 30-40% of lymphocytes in different compartments can be infected with the measles virus.
And did they look in just the blood or also in tissues?
No, that was in tissues. There's a really amazing, an amazing paper in terms of the images that you can get to really get a picture of this in your mind, published in PLOS Pathogens in 2011, I think, by Rick and Paul. And that was something like measles virus lessons learned from the macaque measles experiments or something. And but ultimately, they do necropsy on the primates, and you just see these. You can look at the GFP expression in the gut-associated lymphoid tissue and in the intestines, and look at where all this immunological memory is stored in our intestines, and you can just see these Peyer's patches, which are sort of these patches of immune memory cells and germinal centers, which is where a lot of the immune memory develops, are just completely inundated with virus, and all of those cells will ultimately die off after the infection is cleared.
I think their lips and tongue also glow green.
Oh yeah, exactly. I mean, everywhere where you have it's just
That's
astounding weird to see everywhere the virus goes, and you see it in the skin, probably in tissue-resident memory T cells that are getting infected with the virus. Dendritic cells can get infected, and so that led to this question. Ultimately of, well, if all of these immune memory cells are getting killed off during an infection, do they all come back to normal homeostasis? And do you rebuild your overall repertoire of immune memory after you get over the infection? Or do you kind of recover and have a really skewed immunological memory repertoire after measles? And if that's the case, maybe that could be what explains these long-term benefits of getting rid of measles through vaccination. And so we figured in 2015 that we'd go after and look at this question. And so what we did was we said, okay, if we look at the pre-vaccine era, this is an era of time when nearly 100% of kids were getting measles before their 5th birthday or definitely before their 10th birthday. We would be able to see a signature of that in the population level data if that was really happening, if kids were actually more susceptible to infectious diseases for years after measles. We should be able to look at epidemics of measles as they occurred and just ask simple questions like, do we see excess childhood mortality in the years following an epidemic? And we did. We saw, and that was the 2015 Science paper that showed pretty strong and convincing evidence at a population ecological association level that measles seemed to be associated with 2 to 3 years of excess infectious disease deaths after a big epidemic would sort of go through a population. And so that brings us up to sort of the current study. So all of that work combined said, okay, measles vaccines are introduced, you see reductions in overall mortality. We know that measles virus destroys memory immune cells and lymphocytes. And then we see these large signatures of excess mortality in the wake of a measles epidemic at a population level. Can we actually get at this and measure these questions in individuals? And that required, though, having the right samples. And that brought us to Rick de Swart and the Dutch population, because we really needed to have baseline samples from kids before they get measles and then follow up afterwards in order to evaluate differences. And we also had to have a way to measure the immunological repertoire, which is not a simple thing to do. And Rick, in 2013, had this really great idea. It was a very forward-thinking idea, which was to essentially say, look, in the Netherlands, we have a whole population of people who, unlike a lot of other places in the world, are really happy to participate in scientific endeavors and are very inclined towards science and medicine, but they just don't want to be vaccinated for religious reasons. And
what a unique cohort.
It is. It's a very unique cohort. Really different than some of the rhetoric we see here in the States and why people aren't getting vaccinated. There are people who really have deeply held religious beliefs in terms of not getting vaccinated, but even here, those individuals don't generally tend to want to. They tend to be a little bit more isolated from the scientific community, and so in the Netherlands, they were really happy, and so to participate and so Rick noticed that there was an ongoing measles epidemic that was getting started in the Netherlands, and they had actually just gotten approval to go and get consent from families with in school systems where the kids had less than 20% vaccination rates, and they essentially said, your kid is not vaccinated, your school system has a very low vaccination rate, and measles will probably come through the school system eventually. You can either get vaccinated. Of course, ethically, we want to offer that. But if you're unwilling to, would you at least be willing to allow us to draw a blood sample from your kid? And if your child does get infected with measles, can we come back a few months later and take another blood sample? And these parents agreed. And at the end of the epidemic that happened in 2013, they ended up with about 80 samples that were complete sets, meaning with a blood sample before and after a child had actually been infected and had a full-blown measles infection.
And do you know, is that cohort today, I guess that's, we're 6 years from that, has the vaccination coverage increased? Are these people, if they're more interested in the science, are they hearing the results? Are they willing to change? Do you know about that behavior?
Yeah, I don't personally know. I think I can only speak with some confidence in the answer. And I don't think much has changed. I think it's very, very deeply held beliefs.
Sure.
I was wondering if just the amount of press that this paper got and the other one that came with it, if it might have helped to change anyone's mind in that population. But my guess is probably not, at least not the parents. I don't know. Some of the kids might be, some of them were actually 12 years old, so they're actually getting to be adults now, or, nearing 18 years old. And so they might be feeling differently. I'm not sure. But so that was one thing that we really needed to answer this question: does measles delete immunological memory and cause this immune amnesia? And then the other thing was we really needed a way to measure the immunological repertoire. And not too long after the 2015 Science paper came out, a lot of the reporters and different people were asking me at that time, well, how are you going to measure this? This is all epidemiological studies. What are you going to do to measure it? And so we were thinking, well, maybe B-cell receptor sequencing and things like that. But then it wasn't, I think it was maybe the next week after that paper came out in Science that another paper came out in Science by Steve Elledge's lab. Mm-hmm. And Steve's a geneticist here at Harvard. And he came out with this amazing tool called VirScan. And VirScan is a phage display system which allows us to essentially sort of measure the antibodies across all pathogens and sort of hundreds of thousands of epitopes simultaneously in just a drop of blood. And so I've spent the last few years, when I came up to Harvard to do my residency, I actually started working with Steve to improve the technology and the statistical back end of the technology to make it more quantitative, and ultimately with the idea that we could use this technology once it got to a certain point to ask the question, does measles eliminate immunological memory? And so we ultimately used this phage display VirScan system to measure antibodies against hundreds of thousands of different peptides in these kids' blood before and after they got measles. And we ultimately found that after measles, some of the children were missing up to 70% or so of all of their pre-existing immunological memory or their antibodies against all different infectious diseases after they recovered. So it's really an astounding effect that we've been able to detect here with these children.
And this would include any memory you have from previous vaccination against other pathogens, right?
Yeah, so we think what we really think we're measuring is by measuring antibodies, we're really looking, I think, at a reflection of the bona fide plasma cells in the bone marrow, and so vaccines, just like infections, the memory responses from those end up in memory cells, whether that's in the bone marrow or elsewhere. But we see just sort of an across the board loss of antibodies. And in fact, some of these children had gotten rubella during an outbreak, I think maybe 8 years earlier. And of those kids, we were able to measure their antibodies against rubella, and we found that those were gone. Not completely gone, and that I think has been a misconception. This isn't sort of like a black and white issue in terms of gone, not gone, but a large portion or a large fraction of the antibodies against rubella in these kids was depleted. And so that could suggest that maybe some of those kids might be at increased risk of having another rubella infection. But more importantly, there's lots of different viruses that maybe you don't have develop such strong immunological memory to. And you kind of on the cusp. And so if you lose just a little bit of that memory, you might become susceptible again. And that's kind of what we're thinking is really leading kids to have increased mortality and illness in the years after measles.
Right. And your technology, I mean, you can look at something more than just epitope numbers. You're not just looking at the sequences of the IG region, but you're looking at the strength of the recognition. And
Exactly.
So looking at the relative abundance, it does change I think the gravity of the post-measles immune situation.
Yeah, and we're not actually even looking at the sequences of the B cells. We're really the phage display system. What it is each phage we program them to each project or display a little peptide from a pathogen, and across all of the phage we essentially encode the full genomes, or the full proteomes anyway, of all viruses that can infect humans, as well as a lot of important bacterial proteins. And so these essentially act as little antibody capture devices. Each phage is displaying its own 50 amino acid peptide. And when we mix that library of about 200,000 different phage with a sample of blood, any of the antibodies in that sample of blood will bind to the phage. And if they do bind to the phage, we'll be able to pull those out of solution. And then we look at what phage are pulled out, and the ones that we pull out are essentially the ones that had antibodies bound to them. And we can infer from that exactly what the antibodies were targeting, and exactly what their titer or overall sort of strength of antibody response is. And so it really gives a very robust signal of both the diversity of antibodies that somebody has to a given pathogen, and the titers of each one of those antibodies.
Right. Yeah, and I was thinking about the other paper that we can talk about later that came out around the same time that did look at the sequence level.
Absolutely.
So yep, definitely more robust.
Yeah. So one of the things I was wondering is, a limitation of your approach is you can't get the sequence, right? So you can't figure out to make an antibody, for example, if you find a really good antibody that recognizes a great epitope, you can't really go and clone that antibody because you don't have the sequence. But going the other direction, if you just do the single cell sequencing approach or DNA sequencing approach from the B cells, you can find out the sequence, but you have no idea what it binds. Mm-hmm. Can you envision a situation where you could do both on people and find out if there's a way eventually to connect the two and be able to, can we get to a place, the holy grail would be, could you come up with a sequence and you know what it binds to, so you could make monoclonal antibodies to whatever you want without actually having to, test them empirically?
Yeah, so that is, that's absolutely something that is being done now. Not in my lab, but in Steve Elledge's lab, they're working on these types of questions. And I don't want to say too much about exactly what they're doing, but I do foresee that this is going to be, I don't think anytime soon that particular tech tool will exist. But I think one by one, there are ways now to take really good epitopes and sort of like you can essentially use a really strong epitope to try to pull out different B cells, or maybe tag a B cell or a plasma cell, and then you can maybe sequence. You can siphon off those cells and sequence them, and figure out exactly what the sequence was. And that's actually been done now for a number of different groups to look at the B cell and antibody sequences of various like immunodominant epitopes for flu and things like that. And so I do think there maybe could be a day. My dream would be to create a dictionary so that exactly what you're saying. You can just know the sequences and you know what they're targeting. But there's a big limitation, even if you could do that well. One of the amazing things about looking directly at the antibodies and not the cells is that every single drop of blood that somebody has contains about 10 to the 13th antibodies in it. And, it's an extraordinary amount of antibody in 1 microliter of serum. And what that ultimately means is that just one drop of blood gives you almost a fully representative sample of all the antibodies that you have in your body, which also gives you a very good representative sample of all the plasma cells that are in your bone marrow. And so if you're only sequencing, say, 10,000 B cells from the blood, that might give you a very small slice of your overall immunological repertoire. It might give you a good idea of the overall diversity and these kinds of metrics. But I think I'm just consistently astounded by just how representative every single microliter of serum is that we look at. And I think it because the antibodies you can pack so many into a drop of blood that you can get a representative sample of all of the plasma cells in your bone marrow. I think it really creates a different level of power to evaluate sort of somebody's whole immunological repertoire.
Does that stay static over time, or as you get older, you just get more and more and more and more antibodies per microliter?
Asking a terrific question there. So that's something we're currently working on. And the real question is like, what are the ecological limitations? If you start thinking of the antibody repertoire as an ecological system, what happens to you? And we know that the antibody titers you end up in your blood ends up essentially plateauing out in the first 3 years of life or so. And then maybe you have like a slow creeping up. But then the question is, can you keep building diversity? And can you keep building new memories? And if so, does that come do you have to let some old memories go, or at least some magnitude of those memories go over time? And that's something we're
model, right? Because the expectation is that as you get older, you have a limited ability to make new specificity, so you're more susceptible as you age, right?
Yeah, so that's one model. That's definitely the other model would be that you might start losing them just because you're aging. But exactly, you're up against competing forces if you're trying to push new plasma cells into the bone marrow, or new B cells into your periphery. Exactly. Do you end up pushing up against sort of limitations of the immune system? And on a cursory look, I think that there is evidence that you do start to you sort of top out and plateau out the number of antibodies you can have at a given time in your blood anyway, earlier than you top out the diversity. But ultimately, both will start to plateau, and then you kind of reach this equilibrium, I think is probably what's going to come out of this new area of investigation.
And what do you think about, thinking about, let's say, the gut, which we know has a lot of particularly IgA secreting cells, and if the blood is a representative sample, do you have a bit of skewness for those plasma cells that are more likely to be in the bone marrow compared to those in the guts? And are they trafficking at the same rates out of the gut? And are we missing some gut-related phenomenons?
Yeah, it's a really good question. So if you have some plasma cells that didn't go to the bone marrow, that plasmablasts, for example, that are secreting antibodies elsewhere, I think it's a really good question. Are some of these places in the body, in the context of measles, are they maybe more or less protected from an infection than other cells? And what are we measuring when we're measuring it in the blood? I mean, we know, for example, IgA is, you'd expect that you'd get a different response if you measure IgA in the blood than in mucosal secretions and things like that. So I do think that there might be some element of compartmentalization of some of these cells. But I do think also that the bone marrow, depends on just how detailed the question you want to ask, but I think the bone marrow tends to give a pretty robust readout of sort of the various infections that you've seen
Okay.
Yeah.
Sure.
Is there any way to know if the reduction you see in antibodies across all these pathogens, if that would actually, because it's not zero, right? Mm-hmm. Would that make the kids more susceptible to infection?
Yeah, so that's the big question. Does measles like push some kids below the thresholds of protection from an antibody response, and maybe also T cell responses too, which we haven't measured yet. And I think this study that we did doesn't directly answer that question, which is a limitation of it because our readout is really antibodies and not clinical outcomes. However, we have a paper out in BMJ Open from last year in 2018. I think we published it, and that was following kids using chart review in the THIN database, which is a big UK database of about half of the GPs, half of the general practitioners in the country, and what we found there was we were able to identify about 2,200 kids who got measles in the last 2 decades in the UK. And we followed up their clinical cases for about 5 or 6 years and asked simple questions like, did they end up getting increased utilization of healthcare resources or increased prescriptions of antibiotics over the years after measles? And what we saw in those kids was that there was, for about 5 years after measles, there was an increased rate of prescriptions for antibiotics in these children relative to controls. And there was an increased utilization of healthcare lasting for about 3 years, meaning just the numbers of clinic visits. And so we think that there's, of course, in these population studies, there's always going to be some bias because you'll say, well, these kids got measles because they weren't vaccinated. Are they at extra risk for the infections anyway, relative to controls? And that's a fair critique. But I think what we're really seeing there with all the controls that we did apply, we do see that they do seem to be getting infected at greater numbers in the years after measles. And I think in this case, what we would really like to do next for this study is to take kids after they get measles and ask, did their actual neutralizing titers get reduced?
Yeah.
But that's also that's the sort of placing a pretty narrow, limited view on what antibodies actually do. I mean, the last few years, people like Galit Alter at the Reagan here at Harvard and a number of other people have just opened up this entire new world of really what's the role of an antibody in protection. And neutralization might actually be a small part of that, meaning direct neutralization through antibody binding of key epitopes. Yeah. And a lot more might be elicitation of cell-mediated responses. And so I think to really answer these questions, we kind of need to have more functional-based assays that can maybe ask these questions. Do we actually believe that these kids' immunological repertoire fell below protective thresholds?
I guess you could also model it in your macaque, right? In the paper you did infect some macaques with measles and measured the effect on their antibodies, but I guess you could challenge them to a certain extent, right?
Yeah, and that was actually in the accompanying study. They did that, and they had challenged.
Ferrets,
they had a ferret model, exactly. So it was most of that study was based on B cell sequencing of the naive repertoire, but then they had this really nice ferret model in there, which I thought really drove home the message and worked to corroborate a lot of our population and macaque experiments in our study, which was they gave these ferrets they gave a flu vaccine, and then they gave a similar virus to measles, canine distemper virus, which was a little bit attenuated so that it didn't kill the ferrets. And then they ended up following the ferrets for about 8 weeks or so after the canine distemper virus infection, and then asked what was their response to getting a heterologous flu challenge in this case. And they actually found that the ferrets who had gotten CDV versus those who didn't ended up with reduced protection against that flu challenge. And so I think that these effects are, my guess is that they're real, but I think it's super important to recognize, just based on a lot of the comments I've gotten from people on the paper, a lot of people are saying, saying, oh, well, I got measles when I was young and I never got polio, or never felt
right.
And well, one, there's herd immunity for these things, so that's important.
Right.
But then the other part is that I think some of the really strongly immunizing pathogens and vaccines, you might not actually be throwing these particular responses down to subprotective thresholds so that you would definitely get the infection again. It might.
So we don't even know what the protective threshold is really in a human.
Well, that is the exactly. I couldn't agree more with that statement. Our protective thresholds for a lot of pathogens, like we have these cutoffs, and we say, okay, below 150 international units or whatever it might be, then you need to get a new MMR vaccine. But traditionally, those thresholds have been very poor predictors of protection, and so we really need to. If I were a funding agency and I was putting out a new RFA, I would really push for some research into correlates of protection that go beyond just ELISA titers. And there is definitely a lot of research going on to develop new correlates of protection. But it's really despite the many, many years that the word correlates of protection has been employed and that we've had thresholds, the field is really, really naive and young. And I think there's a tremendous amount more we can do with things like VirScan, where we actually measure individual antibodies. Can we actually get better signatures of a neutralizing titer? Or new technologies that are really taking a subset of sort of cellular functional cells and applying somebody's serum to it and asking, what do those cells do in response? There's lots of new technologies that are being formed that I hope in the coming years will actually start to develop new ideas and kind of a whole new language surrounding this question of what does it mean to be protected, and is there a way to measure it in a sample of blood once you take that blood outside of the context of an active immune system. It's a little bit hard to do.
How does so let me step back a second. When you get an infection, your antibodies to that infection go up and then they go down. So how does convalescence, so sampling later when the antibody concentrations are down, how does that affect how you interpret the results that you get from your studies?
It's a really great, so working with these technologies and these phage display tools and the massive detailed resolution we're getting, it's completely changed how I think about this exact question of what is an antibody titer? What are we actually looking at when we see that the antibodies increase and then decrease and then plateau out? That's sort of normal canonical curve if you look up for an antibody response. And I think what we're seeing is a lot of what's driving that curve, that sort of increase and decrease and flattening out over time into convalescence, is maybe not so much. We usually envision it as sort of you have this big amplification of antibody secreting memory cells, and they all secrete antibodies, and then they contract, and you end up with a smaller number of those antibodies being produced. And we think of that as a homogeneous kind of pool of cells, and they're just all increasing and decreasing proportionally. But I think what might really be going on is you actually have some cells that are going up and kind of a few weeks later or a few months that are going extinct, and some cells that are really getting promoted and then just become long-lived memory cells. So ultimately, an ELISA shows you the average of all these different clones of cells. And what we're finding is that if you actually break out each of those monoclonal antibodies, the dynamics are really quite strikingly different. And it's not just this nice smooth curve that we see on average with an ELISA. It's really much more sharp changes in certain pools, subsets of clones than others.
I think it's important to point out that you also looked and showed that the MMR vaccine, which is an infectious replicating vaccine, doesn't impair the repertoire.
That's important.
Yeah. So we looked a lot at that because we definitely anticipated that would be a question that people would be asking. Of course, everyone's the live viral vaccine. Do we anticipate that it might have the same effect? We saw no effect. And that was really, it fits with decades and decades of experience looking at measles vaccines and just seeing that they're safe and they don't seem to cause any secondary infections. But it was good to have that confirmed in this patient population or in this subset of kids. There was a little, not a hiccup there, but there is a point that we did discuss in the paper that's and that's that the effects are a little bit difficult to look at in that population of kids because the kids who are actually getting vaccinated are about 1 to 2 years old. And during this period of time, their antibody repertoire is really increasing in diversity.
Mm-hmm.
And so over a 6-month period of time, let's say the worst case scenario happened, and there was some reduction in antibody repertoire because of the measles vaccine, which I want to make clear we see no sign of, but let's just hypothetically say it was happening. Over a 6-month sampling period, you might actually have enough compensation just from the speed at which kids are developing their own immunological repertoires at birth, or in early life, that you might actually not be able to detect that so well. And so the actual effect could have been obscured. But that being said, we looked at functional responses as well, and we really saw no evidence. But it was a limitation. It's really hard to look for a reduction in antibodies just because the speed at which these kids are really forming new memories is just amazing.
Mm-hmm. Mm-hmm.
I was thinking about, afterwards, I think in your paper it said, despite the potential benefits of reinstating the antibody repertoire. And I was thinking, oh gosh, well, I mostly think of this as very negative because of the risk, but what are these potential benefits? And I guess thinking about when you are removing past antigenic seniority, you could learn a lot about potentially the impact of broadly reactive antibodies for flu or HIV. And is that something that you're interested in? Have you thought about that?
Yeah, I think that's one of the most interesting questions. We, in an era where we're starting to understand more about things like original antigenic sin,
right?
Yep. Is there actually some? So that's a really interesting interpretation that you have of that sentence. I hadn't anticipated it.
Because I was thinking you actually did not bring up any potential benefits throughout the paper, but it did say despite the potential benefits. I'm like, wow, I don't know what he means there. So it does leave a hole for how is this antibody repertoire going to fill itself back in? You're wiping out. Yeah, like you said, antigenic sin.
Yeah, so in the paper, what I really meant was just if you have your immune memory erased or portions of it erased or diminished, then it's clearly beneficial to rebuild it. That's really all I'm going for there.
Of reinstating the antibody repertoire, not of the fact that, okay, I gotcha.
Right, but
there is,
now that you've said it, it's actually, it's really an interesting point, and I think it will never, I think, be more important than preventing measles.
Right.
But there's these really interesting, if you go back to the 20s and 30s and you look through JAMA at some of these papers, along with all of these like horrible reports of, military of soldiers dying from measles and bacterial infections, you also see scattered in with these measles reports these interesting JAMA case studies, or I don't recall if it was actually called JAMA at the time, but these case studies of individuals with refractory psoriasis and different autoimmune diseases.
Yeah, that's what I was going to go. That's exactly what I was going to ask you about. Yeah, go ahead.
Yeah, and we know now from some work from Tom Kupper's lab and some other places that psoriasis is this tissue-resident memory T-cell mediated process, and so people with psoriasis, they have these autoimmune T cells that just kind of sit there, and you can get them. You can give high dose steroids and different creams, but ultimately they always come back in the same spot. And some people have really refractory problems, and there are these case reports that said, I've had this 13-year-old girl. She's had refractory psoriasis for 4 years, whatever the age groups might be. And then she got measles, and her psoriasis is completely abolished, completely gone, and maybe never even returned, even after a year or two. And there's actually quite a few nephrotic syndrome, and I mean, it's hard to know exactly what they were talking about that long ago, but renal diseases, which seem to be autoimmune in nature, would go away for months or years after a measles infection. So there is some evidence that these autoimmune mediated immune memory processes could have been abrogated by a measles infection. But of course, the risk being that they would also be more likely to die in the meantime from bacterial co-infections and things like that.
Well, I think since you had mentioned that you did work in influenza, I mean, this is not a population that's likely going to be highly HIV infected, so you might not be able to look at that, but you could, thinking about targeting the stalk and trying to create a universal vaccine, it may be an opportunity. But it does lead me to a question about the VerScan, is that if you have a virus like influenza that has, multiple different strains and it lacks cross protection, is a single epitope representative, or are you including multiple? If you're going to weigh these, the diversity of your different viruses that you're trying to target in this technology, are you using more epitopes for flu?
Yeah,
in the phage technology?
We do, and there's a trade-off there, though. We actually have many, so it's a lot of pathogens. For measles, for example, we have 8 full genomes in there. For flu, we have a number of representative strains from each of the different clades. And then we have, for different HAs, we might have HA encoded many different times. And so we can collapse those down if we think that there's cross-reactive antibodies and we're trying to get an accurate measure. We can either collapse those down and treat it as one antibody, or we can really leverage the diversity that we can program into VerScan to really look at different footprints of antibodies. And so there's lots of things that we can do, but that's definitely something that we do. We add many different varieties of the same sort of species of pathogen oftentimes. But then it's really a science that's being worked out now. These are all new technologies and how the data is used and interpreted. Because the trade-off is that if you have many different versions of the same epitope, maybe all differing by a couple amino acids, you might actually dilute out. The antibodies will get spread across all of those, and you won't get a strong signal for any.
Yeah, that's a good point.
And so you can then computationally cluster them together and assume that it's all one, but it muddies the picture a little bit. So it's so really developing this in a logical way is something we focus a lot on, but there's always going to be cost. The higher resolution you get, the more diversity that you might have, but then there's the cost of sort of spreading out the antibodies and losing some signal and things like that.
Sure.
But that's how we deal with some of these complexities, as we do program, and we even do alanine scanning and saturated mutagenesis studies. And this is a lot of Steve Elledge's lab is really focused on the saturated mutagenesis studies, and that's where you take every amino acid in a protein and you flip it to every other amino acid one at a time. And you can really kind of measure the footprint of an antibody, exactly where it binds, what amino acids in a given location will hinder the antibody from binding and what will improve binding. And you can get a lot of information based on those kinds of things. And it's the nice thing about working with phage. You can grow phage to 10 to the 12th PFU per mil, and really get complex libraries. So it's a lot of fun, but it comes with a huge computational cost.
So what's the largest peptide that you can present on one of those phage?
Well, what we present currently with the phage that we have now, they're about 56 amino acids each. But we can we're trying to build it a little bit higher. So it's really a question of what do we want to pay to have the oligos made that we then plug into the phage.
Yeah.
And as you increase the length of the oligos, the price goes up exponentially.
Yeah.
We've been working on different tools and techniques to get more, to get longer peptides into, longer oligos into the phage so that they can express maybe 150 amino acid protein. And then there's even some tricks that you can really push it up even higher. But the price goes way up. And if you're trying to do sort of the whole virome, it gets excessive.
I guess what I was thinking, though, is when you think of an antibody binding a native structure, when you're using 56 or 50 amino acids, you're somewhat limited. It has to pretty much be an epitope that's linear epitope rather than a folded epitope that an antibody might bind. So you are going to lose some of the potential specificities, right? So that's a limitation of your approach.
Yeah, so it's definitely, so if you're trying to say that this is truly comprehensive for all of the antibodies, that would be an inaccurate
Yes.
statement if we were to say that. We know that we're missing a huge number of antibodies that bind to conformational epitopes and polysaccharides and glycated proteins. But what the benefit, especially for a study like this measles study, is that the plasma cells that are going to be producing those antibodies will be they'll all be similar. So we think that the linear antibody repertoire is very representative of the overall repertoire. Mm-hmm. But certainly if we're trying to make the claim that we have a comprehensive measure of all antibodies, I think that's not accurate. It's more of that we have a comprehensive measure of all of the exposures that somebody might have has had, and we can use the linear epitopes, because even with pathogens like flu and dengue and measles, that we know that a lot of the major antibodies that people study are against conformational peptides. Mm-hmm. What we've found, and in some ways has been known for a lot of pathogens as well previously, is that anytime you get infected with any of these pathogens, you get a pretty robust antibody repertoire response to linear peptides as well. And so I think from a sort of prevalence perspective, if we're trying to look at serological responses, we do get a good measure of that.
I wanted to ask you a question that was sent in by a listener who anticipated you being here, I guess. This was Rich. He wrote, I would be interested, on first reading, it appears direct measles infection of B cells leads to damage, but I would be interested in your views on whether this or whether T cell help is involved. Mina et al. Referred to T cells also express SLAM and are infected by measles virus. T cell immunity may be diminished through similar mechanisms, and this could explain the loss of cutaneous tuberculin reactions after measles. Also, do other viruses result in similar downplay of the immune system? HIV comes to mind, but that is more CD4 T cells. EBV may be similar.
Yeah, so that's a loaded question. Absolutely, there are other. So essentially, any of the cells that are expressing the measles virus receptor. So I didn't talk about it before, but the whole reason this happens is because measles binds to this receptor called CD150 or SLAM, and this is it's a cellular marker that's essentially expressed at high concentrations on the surface of memory immune cells and naive B cells and T cells as well. But essentially on lymphocytes, some on NK cells and dendritic cells, some macrophage, but essentially the immune cells in general. And so because that's measles' primary receptor for cellular entry, when it gets into the body, it kind of is squished into this narrow tunnel where the only good cells that it can bind are immune cells. And so that's to say that a lot of other viruses that have the capacity to bind and enter into an immune cell also have the ability to bind a whole slew of other cells as well. Flu, for example, will bind epithelial cells. And so a lot of, even if it can interact with an immune cell, it probably will prefer just to bind to epithelium and degrade the epithelial layers. Measles doesn't have that luxury. It really needs to sort of use this Trojan horse mechanism where it's quite a good mechanism really for itself, where it wants to be picked up by the immune system. It wants to get picked up by these dendritic cells in the lungs and brought into the immune system, where then it can act as a Trojan horse, and it can really enter into the lymph system, start replicating actively in these lymph nodes, and then sort of disseminate. Once it goes through a few cycles, it can disseminate pretty broadly through other immune cells in the gut or in the bone marrow and elsewhere. And that's really distinct. So other viruses don't have that restriction, and so because they can bind to lots of other cell types, they just don't end up living in the lymph system like measles does. But part of that means that measles can also definitely infect T cells, and it can destroy T cells. And some of Rick Deswart's previous papers, I think he had a paper in 2018 in Nature Communications, just looking at the cell subsets that are degraded during a measles infection or diminished. They showed that there were T cell diminishments in the overall numbers of lymphocytes collected in the PBMCs. And then in these macaque models, you see that a large swath of T cells will be infected. So certainly lots of different, really any of the B, T, or plasma cells can all get infected. And that will all be, it's possible that the impacts of the T cell memory pool might be just as important as the B cells and plasma cells. But I think other viruses won't necessarily have the same impact. HIV, I think a lot about making an analogy between HIV and measles. And in a way, you could make the argument that measles is kind of like taking 5 or 10 years of an HIV infection and compressing it down of an untreated HIV infection, compressing it down into a few weeks. And that's the kind of rapid lymphocyte depletion you get from measles. The big difference being that right after you recover from measles, you can start building that back up. Whereas with HIV, you obviously just keep decreasing as long as you're not treated. Right. But I do think that HIV is the other sort of obvious sort of example of a virus that can infect and does diminish immunity. But of course, the mechanism is wholly different.
Anything else, Cindy or Steph?
No, wow. I mean, this was great. I'm really glad that you came on to explain the paper. I mean, we talked about wanting to do this, and having the lead author on has just been really enlightening. I think it's something our listeners will really, really be excited to hear. Now, before you came on, had you heard about Immune?
Yeah, sure.
Really? Oh, that's interesting. Great. That's good news.
Well, mostly, I mean, I think, Vincent, you did, I think it wasn't Immune, but you did something on, I forget if you covered the 2015 paper, or you might have even covered a number of years ago. I published a paper showing that flu mist, an interaction between flu mist vaccine and bacterial co-infections. And you covered maybe both of those, I forget.
Okay.
But ever since those, I've followed some of your various online presences, and Immune was one of those. So, yes.
Well, good. Well, that's good.
In fact, we covered the VirScan number of years ago when it first came out on TWiV, and that was TWiV 342. And I have to tell you, the title of that episode was Public Epitope Number 1.
Good. Good.
I like that.
And more recently, a cool study came out of the Lipkin lab. They used VirScan to look in cerebrospinal fluid of kids with acute flaccid myelitis, which is associated with enterovirus 68 infection. And very few people have been able to isolate virus from CSF, but you can find antibodies in CSF using VirScan. Yep. And I think that's a really cool, as you say, you can get so deep with this. And I envision one day, instead of just looking at neutralization for one virus, they're going to use VirScan to just, in a short amount of time, look at everything.
Well, that's what I'm working on now.
But yeah,
we'll have to follow your progress.
It'll be part of your medical history. You're going to have a record of everything, and it's going to be mechanized so that it'll be easy, just like any other diagnostic test.
Yeah, exactly. One thing I do a lot of serious surveillance as well, and work. I've been trying to build up the technology to use as a public health tool. Besides really understanding basic biology questions, pathogenesis, it's to really use this as a serological surveillance for outbreak detection. And the big limiting feature at the moment, or factor, is that it's a pretty manual assay still. We do use liquid handling robots to run it, but there's a number of washes and things like that. And and so getting it into, what I would really like to do is make it into sort of a cartridge-based thing, kind of like a Cepheid gene expert type of approach. I'm also, because I'm a clinical pathologist, I work in the lab all the time in the hospital and oversee parts of that. And it'd be great if we could get this tool into a format that you could send it to anywhere in the world, and with a little kit, and they'd be able to run it in a little plastic sort of compartmentalized process. I'd like to see it there in a number of years.
Very good. Michael Mina is at Harvard University. On Twitter, Michael Mina _lab. Go follow him. And Michael, thank you so much for joining us.
Well, thanks so much. This has been a real pleasure. I've very much enjoyed it.
Great.
Oh, great.
And good luck with VirScan.
Thanks so much.
All right, bye-bye.
Bye.
Take care.
What does the second paper do that's different? Why don't we quickly...
Yep. So I will admit, I read both papers because I thought it was really fascinating when I was preparing for this, and I was just simply blown away by the VIR-scan approach.
Yeah.
That I read the other paper second, and it probably would have been better to read it the reverse way, because the second paper really isn't. I mean, it has a lot of stuff, and it's really fascinating. And I think the advantage that they have is that they go about it from completely the opposite direction.
Right.
So what Michael was saying is that the VIR-scan, you take a droplet of blood and you're using basically like a bunch of different flags, and you're trying to pull out all of the antibodies that recognize those flags, and then you can see what they recognize because you just sequence the crap out of everything that comes out of those phage. Well, this is completely the opposite side of it. So what they did here was they took the same exact idea. They have similar cohort, they have different sets of controls, but it's basically the same idea. People who are in a highly susceptible population but who are unimmunized, and bleeding them, and then bleeding them again if after they develop a rash or if they don't develop a rash. And then binning them into, seroconverted or non-seroconverted. And they take their B cells and they so the limitation is it's blood, right? And so we talked about that a lot of those long-term memory B cells will be either in the gut or in the bone marrow. So you're going to completely miss them, which is a huge limitation of the paper and an advantage of Michael's approach. But they just take and they purify the B cells, and they do flow sorting. So they get their purified population, quote unquote, which there's always limitations to that. And then they do single cell sequencing. And so they sequence the B cell receptor, which is the antibody region. And then they can analyze, using similar big data approaches of the repertoire of B cell receptor sequences before and after, and they find the same kind of idea. So once you have a measles infection, the B cell sequences go away. They go down, and you lose them, and they don't come back. And you start to develop new ones. So Michael was talking about that once you have measles and you get this amnesia, then you have to rebuild your immune system and the memory. And what this group shows is that those new B cell sequences that come up are different. So when you first generate B cell receptor sequences during fetal development, and then as you get exposed and you start messing with the sequences, they have a certain characteristic, a certain length, and certain types of sequences are preferred, organizations. And once you have measles, when they rebound, they actually look different. And one of the differences is in this area called the CDR3, or complementarity determining region 3, which is the part of the antibody that actually contacts the epitope. And they find that the length and the diversity of these CDR3 regions are very different once you've had a measles infection, suggesting that the actual repertoire that's rebuilt is fundamentally different.
And what's the functional consequences? Do we know?
So I mean, they argue the same thing that Michael was arguing, that it's going to change your susceptibility to infection because you've lost different repertoires of B cells, and that when you rebuild it, it could be looking different. And so one of the ideas is the older you are when you get measles, the more you wipe out, and when you rebuild the repertoires, it's going to be different. And so you might have a different susceptibility than someone who had it earlier in life, but that's all conjecture.
Yeah.
And I think that's why they had to do. They did the ferret?
Yeah, they did.
They did the ferret. Right. They did, and that was because you're right. You don't really have a functional readout other than to say, okay, the repertoire is more immature looking, and like Cindy said, you can draw conclusions. But the ferret model does help this paper to drive home their sequencing data.
Yeah, and I'll tell you, if the VIR-scan paper was not out there, and I think he had a very good argument, this is the state of the art. I mean, doing this B cell single cell sequencing and looking at all these B cell sequences, if you saw that on its own, you'd be like, oh, wow, this is cool. So, I mean, this is actually a fundamentally important paper. It's just when you now put it in contrast to this other one that they're looking at the actual antibodies binding to epitopes, and you're like, oh, well, crap, that's better.
It's a good combination.
Right? Yeah, they really, I think we can't underestimate the impact of both coming out at the same time.
Yes.
Because showing that the B cell receptor sequences are different and the antibodies actually recognize different things, and you lose the binding capacity for all these different pathogens, say that this is really real. This is like, when you have epidemiology, I have this veterinary course that we teach, and part of it is epidemiology, and I'm learning that on the side as we're teaching this whole course. I don't actually teach that part, but I'm involved in all of that and writing quiz questions and test questions. But anyway, learning this sensitivity and specificity and testing in series and parallel. So if you know any of this, if you have two different tests, you can say if one or the other is positive, it's a positive result, right? Or you can say they both have to be positive to believe it. And it changes your willingness to say, oh, this could be an artifact or a false positive. But I think the take home with what I'm saying here is that whether you do that independently or together, both of these tell you the same thing from two completely different approaches. And so the likelihood that this is just something weird that happened is basically nil. So this is really compelling that measles infection is destroying the capacity of the immune system to have memory, and it has to all be rebuilt.
So let me ask you, if you did either one of these two studies with HIV-infected patients, what would you see? Because the target cell is a CD4 T cell, not a B cell, right? But you need those to make antibodies, right?
Yes, you do.
So what would you see? Similar, it just would take longer?
I would think yes.
Yeah.
Just because you lose T cell specificities over time, and so I think what you'll have that's different from this is that you're probably not going to lose your long-term memory B cells. What you're going to lose is the capacity to generate new specificities. So I think that the outcome of the study would be fundamentally different.
So you would not see a decrease in the antibodies like they did in these studies. Right?
I don't, unless HIV goes and infects those cells, which we don't think they would.
Yeah.
Or the
pediatric infections
to keep them alive. Okay, pediatric infections would be a different thing.
There's like 160,000 new pediatric cases a year. Now, because of ART, you're going to be able to control that, but a lot of areas there's not, a lot of people don't stay on ART. So I think those children, you could look and see if that is true.
Mm-hmm.
Yep.
So the other paper we're talking about was published in Science Immunology, and which
is a newer journal.
It's a newer journal, yeah, because we can't have too many journals, I guess. Incomplete genetic reconstitution of B cell pools contributes to prolonged immunosuppression after measles. And the first author is Velislava Petrova, and the last author is Colin Russell. And on this paper, we have also Rick de Swart, who has the cohort in the Netherlands.
That's
and
right.
We also have Antonio Lanzavecchia, which I just want to say because I can pronounce his name, and he's a famous immunologist.
Yes. Yep.
Veronica von Messling, I happen to know she's a vet who did the ferret studies, I'm sure.
Yeah, she's fantastic. I've met her. I like her.
And this is a European consortium, UK, Netherlands, Germany, and so forth. Anyway, two cool studies. Very neat.
Very, very cool.
Yeah.
And,
it's great to have Michael on.
Michael's very good. He knows his stuff and can explain it really well.
He really could. I was worried. This is one of our first interviews. The only other one we did was when we were at ASV last year, right? But I was like, oh, is he going to be able to speak so our listeners can understand? But he did a fantastic job.
Great,
really did.
Yeah. For some reason, the microbe TV is a selection process. You just do well. This Rich, who wrote us this letter.
Yeah, who's Rich?
He's a listener.
Oh, I thought maybe you're going to identify him as something.
No.
I don't know.
He wrote, as an aside, I saw Antonio Lanzavecchia was co-author. I studied immunology as part of my degree in the late 80s and was impressed at his output then in terms of the number and breadth of the papers he was publishing.
Yeah.
Many thanks, and can I say keep up the podcasting? I wish you were weekly rather than monthly.
Maybe if I have my own office someday, I can do that.
You get your assistant professor job, and you get your first grant, and then we'll talk about
and then we'll go weekly.
Hopefully, I'll be around by then, right?
Oh, you'll be around.
We could do twice a month, right?
We could.
Would be
we
better.
Could try. Yeah.
We don't have to
I
do
think
it now.
coordinating our schedules is hard enough as it is. Oh, I definitely think there's enough immunology out there to do it every week. There's no doubt about that. I struggled to pick. There were so many good papers, and I was like, oh, I just can't not do this one because it's just so great.
Timely.
Very much, yeah. It's very cool. But sadly, as he said, it probably won't change people's minds who do not want to be vaccinated, which is very sad.
It may, though. I'm going to say.
You're going to be optimistic because that's who you are. Yeah.
I'm going to say.
And I are very pessimistic about that.
I know, but maybe I just need, to be beaten down more in this arena. But maybe this compared in talking to more people, I can try to change their minds.
Oh, I have one last question. So if you did this study with, you took a bunch of people and you did them at, 1 year of age, 10, 20, 30, 40, 50, 60, 70, 80, and you took that whole series and did this study, what would you see? Would they increase or decrease?
Yeah, so that's what I was asking him about when we were talking about aging. I would put money on that what's going to happen is for the first 5, based on having kids and when they start to not be sick all the time, first 5 years is just going to explode in the number of specificities, and that the titer will slowly rise until it reaches whatever that maximum is. And I think how the immune system determines those maximums and maintains that with T cells and other cells in the blood and antibody. I don't know how that works, but it's fascinating. But anyway, once it reaches that maximum, I think you're going to stay around that. But what you'll start to do is have more clonality and less diversity. And that it will just continue over time as you have infections. Probably flu is going to be one of them because we always, many of us, I don't know how many, many of us expose ourselves every year by vaccination or whatever. So you'll start to have more and more and more skewed, and probably around 60, you're going to start to have more and more against varicella, right? Because we get the shingles vaccine. And so it'll start to skew, and you'll have less new specificities, and you'll start to lose specificities. And so you'll just get the same titer, but fewer things that are recognized. That's my guess.
Yeah.
Oh, I'm sure someone will do it at some point. I mean, there are all kinds of applications. We'd be really curious just to get a base, as I said, do clinical samples and do this for everyone. And maybe you have some weird skewing that correlates with some condition you have, right?
Oh, that would be fascinating.
That would be very cool.
All it's limited by is, how many of these cartridges or whatever, once he makes them, you can make.
Yeah,
because it's a drop of blood. You could screen hundreds of thousands of people, and the amount of data that you could collect and start to bin, you could start to profile people that way and find out what kind of diseases they have based on their repertoires of antibodies, probably.
A drop, it's really, you don't need a whole tube.
No.
But they will take a tube anyway and throw most of it away. So we have a book to give away. It's a copy of this immunology lab manual. So we had 14 people write in. And let's see. We had Annette, who is a senior at the University of Washington, taking her first immunology course and loving it.
Oh, that's great.
Finding this podcast has been wonderful. Is enhancing my learning and understanding of the concepts presented in class. I'm planning on continuing into a master's in epidemiology and perhaps a PhD in immunology.
Go for it.
Yes, Annette, go.
Nick writes, hi, immune hosts and associated microbes. I'm a microbe PhD at UC Davis. I love all the Microbe TV podcasts. I'm currently reading papers while drinking coffee out of my TWiV mug. I'd like to win the manual. Thank you for all your hard work. Gabe. Recently found your podcast. Absolutely love listening to you guys. Keep up the good work. Much love from Sweden. Cool. Larry writes, I love this podcast. I'm listening to Twin right now. Caleb, I love all your podcasts. You do an amazing job communicating complex concepts. Jason loves the podcast. Thank you so much for helping keep my interest in learning about immunology. I have a double bachelor's in biomedical science and chemistry. Currently working in manufacturing for a T-cell therapy treatment. Listening to your podcast helps me on my 3-hour commute in LA.
Whoa.
Oh, man.
I would literally, I can't even fathom 3
Steph,
hours.
you're going there next.
What? To LA?
3-hour commute. That's it. By the way, the weather is nice and windy, starting to get to about 70 during the day. What is this? 70?
Delightful.
Well, I guess that's the benefit. Yes, 3-hour commute. It's 70.
Ed writes, "Hi doctors, I'm an avid listener in California, currently basking in mid-70s, which provide ideal for my tennis and golf pursuits. Many years ago, I triggered a gluten intolerance after enjoying a large plate of linguine with clams. I'm so sorry. It's such a good piece of food, linguine with clams. Seems like a lactose intolerance appeared shortly after. Your wonderful podcast provides me with a better understanding of the complexities of the immune system. Eventually, there may be solutions for my problem similar to what I've been reading about the peanut allergy. Keep up the great work you're doing. Very much appreciated." And Trudy also wrote in. Kim wrote in, "I reached out a while back. Although it feels like all I do is flow cytometry, we can always improve. Also, I reached out a while back indicating that I would do transcripts. A couple of episodes ago, Vincent mentioned he would email me, but I never heard, and I figured I'd just get started, as I've been looking forward to giving all the episodes another listening. Started yesterday, see attached Word document, and I anticipate I'll do about one a week, so we can catch up in the few months."
Cool. Thank you.
All right, so Kim is
Nice.
Brienne Barker's sister.
Oh, yeah. Right.
Oh,
And Kim is a graduate student.
cool.
And we did actually get our first transcript. I posted it. It's on the first episode. So if you
Well, that's good.
that's very nice.
Well, I
Thank
guess
you,
we
Kim.
can't get to more than once a month until she catches up, because then otherwise we'll get so far
Yeah,
ahead
really.
we'll never get there.
Oh, she won't do once a week. It's just too much. She's a grad
It
student.
is.
No way.
I
No way.
know. No.
Danielle wrote in, Cassiani, "I love listening to your show. An undergraduate finishing my bachelor's in biology, planning to earn my master's and PhD in immunology. Your show is inspiring. I learned so much. Keep the podcast flowing. I wish there could be episodes more often. Great work." And then one from Delbert, longtime listener here. "I've listened to every episode of Immune, every episode of TWiV. Love both immunology and parasitology. I listen to the other podcasts in the series too, but not as religiously. These are some of the best podcasts out there. Keep up the good work. In fact, your podcast inspired me to start my own podcast a while back. We aim to debunk science myths. It's so important to educate the public about science." And Cindy, yes, I am the Delbert for Dr. Denker's lab at Cornell. I miss those days dearly. Happy times indeed.
Yes, I remember Delbert.
Oh, that's great. Is that?
Yeah,
who you were
I
trying to
saw
think
that it
of?
was Delbert, and I was like, I only know one person named Delbert. And I wonder if that's who that is. And it is. Hi, Delbert. We miss you too.
Emil writes, "I start this."
Oh, you did miss Anthony? Did you?
Yeah, Anthony wrote in. Please enter my email.
Okay.
Emil, I'll start this email the microbe TV way. It's raining here in Canada. After a few weeks of cold weather and snow, it's probably going to freeze over in the next few days so we can skate down the street. Meaning to write for you a while. I guess winning a book is a good incentive to get over the hurdle of sending praises to strangers. We're not strangers. We're your friends.
Strangers, we're family.
Yeah, of course.
This podcast is really awesome. Thank you, Vincent, for keeping it all together. I felt safe when you said, I'll be doing this forever. Never stop, never stopping. The things I learned listening to your podcast helped me get a promotion and set me on a path. I'll be forever grateful. Great.
Wow.
Have a good day. P.S. We can hear your TV or radio in the background. There's no TV or radio playing here. I don't know what you're listening to.
What's interesting, and I have actually, it brings up, there was a couple episodes years ago that I could hear something. And if you listen to Paula, Stephen, or Stefan from the TWiV episode on the economics of science, you can very subtly hear music in the background only, I think, when Vincent, it's only when one person talks. And it makes me feel like you're picking up somebody else's, I don't know, somebody else's music. So listen to that, Vincent, and see if you can pick that up.
Yeah, because I didn't, there was nothing. It was in her office and there was nothing playing, no TV, no radio. It could be picking up some interference. But,
I feel like that's it. It's very subtle. It's really hard, but you can hear it.
And because I'm deaf, I don't hear it. So
but I don't know how you solve that problem. I don't know.
We have great equipment thanks to Vincent.
It's hard to.
We have awesome audio.
I don't know which episode Emile is writing about.
Maybe Emile, you can let us know which one, and we'll go back and listen and see if we can figure out what it is.
Yeah, maybe. Yeah.
Because I'm pretty anal about keeping the quality of the sound good. So I'm kind of sad that you're hearing it hopefully
I don't honestly, and maybe it'd be interesting if where he heard it was the same one I heard it, because I don't hear it every time for sure. It was just this one time I can recall.
Well, rest assured, we do not record with music or TV.
Elevator music playing on.
No. All right, so let's pick a random number. We had 14 entries. 1 to
Drum roll,
14.
please.
Drum roll. Generate number between 1 and 14. 54. No, that can't be. No. Let me do it again. Number 4.
Okay.
Number 4.
Who's
Larry.
Larry. I do.
Great.
Okay, Larry, you're the winner. So what you need to do is just email me your address. If you're in the U.S., no problem. That's all I need. If you're overseas, I'll also need a phone. Larry is the winner.
I love doing these, by the way. I like hearing.
You do?
It's fun.
Yeah, it also gets people to write in. And
that's right.
I love hearing what people are doing, right?
Yes. Yeah.
It's very cool. All right, that is TWiV. This is funny. November 26th. It's Immune 26. 26. And you can find these show notes at microbe.tv/immune. You can send your questions to us. Or if you don't even need to send a question, you could just say, hi, I am doing this, and I like listening to you while I do it. Immune at microbe.tv. If you really like what we do, consider supporting us. You could give us $1 a month. It's not a lot of money. $12 a year. Not much for you, but it would help us a lot, because if all of you did that, it would help us with our expenses. Microbe.tv/contribute. Things like Patreon and PayPal will let you set up payments like that. And some people say, well, I don't like to set up monthly payments. Well, you could just give us like 2 years worth of, that would be $24, or 1 year. Some people do that.
What a bargain. $24.
I mean, you get all the podcasts for free. That's because everyone does it for free. And I'm really
That's right.
I'm very grateful that everyone on every show does this. I could not do it on my own, obviously. I could just do TWiV maybe, but it wouldn't be as fun. It's more fun when you hear what other people have to say. So our guest today, as you heard, was Michael Mina. Again, Twitter, Michael_Mina_lab. Cindy Leifer is at Cornell University. Over on Twitter, Cindy Leifer. Thanks, Cindy.
Thank you.
Steph Langel doesn't have a 3-hour commute because she's at Duke University. And on Twitter, she's Stephanie Langel. Thank you, Steph.
Yeah, thanks. This is wonderful.
I'm Vincent Racaniello. You can find me at virology.ws, PROFVRR on Twitter. Music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.